Hello and welcome. Today we're going to explore official standards for pharmaceutical chemicals and formulated products with a focus on the United States Pharmacopoeia. My name is Ken Ngoke and I will be guiding you through this important topic. These official standards are essentially the quality rule book that ensures our medicines are safe, effective, and of consistent high quality. We'll cover what pharmacopoeia standards are, how the USP works alongside regulators, and why all of this matters to you as future pharmacists. Okay, so pharmacopoeia standards, as you might see, uh, when you ask what are pharmacopoeia standards, you see, they, they are the official quality benchmarks for medicines and their ingredients. Think of a pharmacopoeia as a big book of quality recipes and tests for drugs. In the United States, for instance, the primary compendium is the United States Pharmacopoeia and National Formulary. And this is an official reference that contains monographs. And what are monographs? They are detailed standard documents for drug substances, including active pharmaceutical ingredients, dosage forms, that are the finished products, tablets, injections, excipients like inactive ingredients, including starch, binders, starch or binders sometimes, or fillers, or even dietary supplements. Each monograph sets requirements for things like identity, strength, purity, and potency of specific substances or product. Why are these substances so important? Because they ensure that no matter who manufactures a drug, it meets the same minimum quality criteria. So, in fact, in many countries, these standards have the force of law. In the US, for example, the Food and Drug and Cosmetic Act says that if a drug is listed in the USPNF, it must comply with the USPNF standards to avoid being considered adulterated. This means a company can't just claim their product is USP grade or put USP on the label unless it truly meets all the tests and specifications in the monograph. If it fails even one required test, legally that product isn't of acceptable quality. Regulatory agencies like US Food and Drug Administration and the equivalent agencies such as NAFDAC in Nigeria enforce these standards by ensuring manufacturers adhere to them. So, pharmacopoeia standards are not just academic ideals. They are practical rules that protect patients by guaranteeing consistency. Whenever you see USP on a medication bottle, you can have confidence that the product has been made and tested according to these stringent official standards. Just know that, like I mentioned earlier, the other pharmacopoeias, the European pharmacopoeia, the British pharmacopoeia, the Japanese pharmacopoeia, and so on and so forth. So the role of USP and regulation. Remember, we are looking at USP on behalf of other pharmacopoeias. Okay, and the USP standards are enshrined in US law, F&DC law, and I mentioned it in the last paragraph. So let's take a bit more about, talk a bit more about the USP itself and its role in regulation. The USP is actually a non-governmental, non-profit scientific organization. I'm not sure you know about this, but you have to know it today. It's not a government, it's a non-governmental body, but it has power because it has power has been given to it by the public, and it holds the power in trust. It's been around for over 200 years, setting drug standards since the early 19th century. Even though USP is not a government agency, its standards are recognized by US law and many other jurisdictions. So this creates an interesting dynamic. USP sets standards, but regulatory agencies enforce them. And these regulatory agencies are government agencies. Okay. So we talk about FDA in the US and we talk about NAFDAC in Nigeria. For instance, the USP publishes the tests and acceptable criteria for a drug's quality, and then the FDA or NAFDAC will check that pharmaceutical companies are following those standards for any drug that's supposed to meet USP specifications. So official standards ensure consistency and safety. That's about the public trust I mentioned earlier on. The USP operates through a public process, collaborating with the experts, pharmacists, chemists, physicians, and regulators worldwide to keep standards up to date. The standards are considered public standards, meaning they aren't proprietary to any one manufacturer. They're openly available benchmarks that any lab can use to test a product. Many countries either adopt the USPNF standards or use them as a supplement to their own local pharmacopoeias. Remember I mentioned that we have the British pharmacopoeia, the European pharmacopoeia, Japanese pharmacopoeia. It's common to see pharmaceutical ingredients labeled as USPEP grade or PPJP grade, indicating they comply with multiple pharmacopoeia standards. So in summary, the USP's role is to establish these quality standards, and regulators' role is to enforce them. If a manufacturer doesn't follow the compendial standards, for instance, their product fails a USP test, regulators can take action, like preventing the product's sale or issuing recalls because the product would be considered substandard or adulterated. This partnership between a private standard setting body and government enforcement ensures that standards maintain scientific rigor while having legal teeth. As pharmacy students, it's useful to realize that USP standards are pervasive. Whether your practice is in the industry or in a hospital or you are a community pharmacist, these official standards are behind the scenes guaranteeing that the medications on the shelf have the quality they should. When you talk about the process Let me just come back a bit and talk about the process of establishing, um, the USP standards. And those standards, you know, are like the things you see in the monograph, as we're going to talk later about the monographs. The monographs are established if, like, a company brings up um, a product they want to market. They prepare the monograph, send it to the USP, the scientific, committee of the USP or scientific staff of the USP will review the monograph, and after reviewing the monograph, they will conduct some tests to confirm that the results they saw in the monograph or the proposed monograph from the manufacturer tallies. There's kind of reproducibility. So when they are done, they will send it as a proposed monograph to committee of experts, who also reviews and then publishes it in the pharmaceutical forum. And that forum, everybody is allowed: pharmacists, doctors, journalists, whoever that has a comment to make will make all the comment, and that will be reviewed. And when the comments are reviewed, the COE will take it and then recommend for the monograph to be accepted. So that makes it a public document, and there is a kind of public trust. A public energy is put into the development of the monograph or the USP standards and all that. We have standards already, so the monographs are judged against the standards we already have. And generally, if you have what you think is the characteristics of your new product, they are tested and confirmed before it's adopted, and that becomes a standard as well for that product. So let's look at the USPNF and other pharmacopoeia. So like I said earlier on, I'm just using the USPNF as a focus, as a representative of other pharmacopoeia. Virtually every region or country has its own official pharmacopoeia, official compendium of drug standards. I don't know of anyone from Africa or Nigeria, but I know that Europe has one, Japan has, US has, then you have the one for Britain, and these are the very popular ones. They have the International Pharmacopoeia, for instance. You can think of what I want. The Chinese definitely has their own pharmacopoeia, but definitely it's going to be in their language, which probably might be difficult for everyone to understand. So a lot of content in this are harmonized or they overlap. We have the Indian pharmacopoeia in addition, and then let's just push it that, like I was mentioning. And as I said, these pharmacopoeia serve the same purpose in their respective regions, ensuring the medicines meet predefined quality criteria. In practice, many pharmaceutical substances are manufactured to comply with multiple pharmacopoeias. You might have seen chemical bottles labeled something like USP grade, meaning the product meets both the USP NF specifications and the European Pharmacopoeia specifications. Global manufacturers often do this to be capable of marketing their ingredients or products in different countries without having to make multiple versions or trying to make new registrations, going through the rigors of proving everything. The moment they are able to get these things using the standards, the process goes very easy. There are also a lot of collaborations among pharmacopoeia nowadays. Through international forums, pharmacopoeia organizations strive to harmonize standards so that, for example, the test for a particular drug's purity might be the same whether you look in the USP or the EP. This reduces duplication of effort and test for companies and ensures consistent quality worldwide. So you have international harmonization committee, that sees to some of these things. For the purpose of our discussion, we'll focus primarily on the USPNF as an example of official standards. Just keep in mind that the concept generally apply to any pharmacopoeia standard, whether it's USP, BP, EP, Indian pharmacopoeia, or IP, the international pharmacopoeia. The goal is the same: to provide a quality blueprint for medicines. And speaking of blueprints, let's move on to USP monograph, which are the central pieces of this standard, essentially the blueprint for individual drugs. I mentioned this earlier on. So USP monograph, the blueprint for quality. What is even in a monograph? So at the heart of the USPNF is the monograph. A USP monograph is like an official dossier or blueprint for the quality of a specific drug, substance, or product. It defines what that substance is and how to verify its quality through various tests. Every official drug, including excipients and dosage forms, will have a monograph in the USPNF titled by the drug's official name. For example, there is a monograph for aspirin, for the raw chemical, and another for aspirin tablet, for the finished product. So aspirin, the raw chemical, acetylsalicylic acid, or the aspirin, the tablet. There are monographs for things like amoxicillin powder for oral suspension, metformin hydrochloride, glycerin, and so forth. The paracetamol, acetaminophen, covering both the active ingredient and the formulated products. There are some variations in something like not less than, not more than range, while some for the active, the raw chemical might be between 98 and 102%. The formulated product can be between 90 and 110 or 90 and 120, depending on the product we're looking at. So I wanted to look at the monograph as the quality recipe or contract. It usually starts by naming the substance and providing a definition and a list of all the tests and criteria that substance must meet. If a manufacturer claims that their product is, say, ibuprofen USP, then that product must meet all the requirements in the USP ibuprofen monograph. So we're talking about the definition and identity. So the monograph defines the substance, required identity criteria, the name of the drug, the drug name, which is the official title, purity and potency tests, assay verifies active ingredient amount. I mentioned something like that, 90 to 102. Another requirement, specific tests, storage, labeling, physical constants like melting point, optical rotation. Okay, so if it fails any single requirement, say the assay shows only 95% of labeled ibuprofen, but the monograph demands at least 98%, then the batch is not acceptable as per official standards. So even if it's just 97% and after repeated tests, it's still 97%, it's still not acceptable. So the 97 to 98% we mentioned there is like the not less than, the lower limit, and the upper limit is the not more than. So the monograph ensures identity, confirms that the substance is what it is supposed to be. It confirms potency, the strength amount of the active ingredient. You have to make sure that it's correct. Purity, you want to be sure it's not contaminated with too much of any impurity or foreign substances. Some impurity are allowed, but there are ranges that you will be allowed to have. And that's why you do assay. You do assay for the main content and you do assay for impurities. There could be impurities that are degradation products, which is what is measured if you want to revalidate a medication that has passed its shelf life. So you look at the active ingredient, the API, and see what's the percentage of the API, and then look at the percentage of the degradation product. And if the degradation product is one that is dangerous, you also be very careful whether it has reached the limit that is not allowed. It's part of the reason why we do revalidation. So if it's not contaminated with too much of any impurity or foreign substances, then it can pass, but you have limits for each one. And often other qualities like the dissolution, performance for the tablets, or proper labeling of content. So the monograph is very comprehensive and it tells you what you have to do and what has to be on the labels, the packaging has to be. It's also worth noting that monographs often reference USP general chapters for specific methods. So what this means is that if you are looking at the monograph, it will not always describe, maybe for test or an assay method, but it will not always describe all of it, for you. It will always refer you to where the general method has been explained. Let me say chromatography, chapter 197 or page 197. So rather than describing, how to do a chromatography test from scratch in every monograph, the monograph will reference general methods, 621 for chromatography, 731 for loss on drying, etc. This keeps the monograph text concise. I want to believe that it's concise, but it's not always concise. But you can imagine how big it will be, if you have to do the description for every item. So it means that a lab using the monograph will need to refer to those general methods for exact procedure. In essence, a USP monograph and the general test chapters together tell you exactly how to test a drug or what results are acceptable. And in practice, quality products or quality products must have to comply with, like I said earlier on, with every requirement specified. And quality control, I follow these instructions to the letter to verify each batch of a drug. We'll soon break down what specific section you can find in a typical monograph, but the big picture is a monograph is the official quality standard for a substance. truly a blueprint or checklist that ensures any material labeled with that drug name is of high and consistent quality, and it is what is said it is in it. There are other requirements, specific tests such as storage condition. I mentioned the labeling, physical constants, melting point of decorrotation for chiral compounds, the storage condition. This is very, very important, and all that. These are things that you have to conform to the requirements. It has to be exactly what is stated or within the range that is allowed. So like I said earlier, um, from where I stopped before I was interrupted. You talk about the purity and potency of monographs. Now, the assay is a quantitative test to determine how much of the active ingredient is present. For a pure drug substance, this is often in the range of 100%, near 100%. It's always like 90 to 102. I talked about this before for the raw substance. Meaning, if you acid the powder, it should contain essentially all active drug with only minimal allowable deviation. For a finished product like tablet, the assay might ensure each tablet has between 90 to 110 of the labeled content, accounting for manufacturing variability. Sometimes, depending on the product, something like amoxicillin trihydrate might have up to 120 because of, as the upper level, because of its high degradability. And some products like that are packaged with desiccants. So this ensures that patients get the right dose. Then there are impurity tests. I've talked about this before. This could be specific tests for certain, likely impurities, or general tests like related substances by HPLC limits on heavy metals, residual solvents, especially if the products are produced through synthetic procedure or extraction. So extraction solvent might be there, or maybe some side reactions, the product of side reactions during the synthesis might also be there. So you want to know the limit that is allowed, how the purification process was how efficient was the purification process. So impurity tests set the maximum allowable levels of degradation products, side products, or contaminants. For instance, a monograph might say no single unknown impurity greater than 0.5% and total impurities not more than 2% as criteria. These purity tests guide against contamination and degradation, ensuring the product isn't carrying potentially harmful or excessive impurities. Other requirements beyond identity, potency, and purity that monographs often list, are specific to the substances. This might include tests like melting point range, optical rotation for chiral compounds, pH of solution, or a test for clarity of solution. So any test that doesn't fit neatly under assay or impurity, but is important for that particular material's quality, is listed in other requirements, and these things are specific. There are also often instructions on packaging and storage. The monograph may specify that the substance should be stored in a tight, light-resistant container, or kept at certain temperatures, used within a certain time after opening if it's prone to decomposition. Labeling requirements may be included as well. For example, if a drug is a salt, the label might need to state the percentage of the active base. Let's say, for instance, you can say each tablet contains 100 milligram of drug, say hydrochloride, drug X hydrochloride, equivalent to 80 milligram of drug base. Let me repeat that. If you can say this contains 100 milligram of drug X hydrochloride, equivalent to 80 milligram of drug X base. Or an injection that is meant for single use might need to be labeled single dose. The monograph will list the necessary reference standards. So you could also say like USP drug X reference standard that are to be used in performing the test. I can explain there has to be in NAFDAC, they call it primary standards, but it's the reference standard required. These are official samples of the drug or impurities that USP provides so that labs can compare their test result to a trusted standard. So that is why the NAFDACs or the equivalent of NAFDACs in different countries collaborate with USP so that they will be able to get these official standards or the primary standard. So all these elements together form the monograph. In short, monograph defines the substance and then lays out how to identify it, how to measure its potency, what the limits of impurities are, and any special tests or handling requirements. Packaging, handling is like packaging. If a product meets everything in this list, it's good to go. If it fails any part, it's not considered of acceptable quality. So we're going to break down these sections one by one to make sure each is clear. Now that we know what a monograph is and what is in a monograph, so how do we use it in practice? Imagine you work in a pharmaceutical company's quality control lab or a compounding pharmacy in a hospital. When you receive a batch of a raw drug powder, the API or the active pharmaceutical ingredient, you will pull out the USP monograph for what the substance is and run all the tests listed there on a sample from the batch. In fact, this is the kind of thing you're supposed to do when you receive products in a hospital. If you have a compounding lab, when you receive new products from any batch, any new batch, you're supposed to run all of them to see that what you have is of good quality, comparing to the monograph that you've had or you have in your store. So let's say you receive a drum of aspirin powder The QC lab will perform the identification tests, perhaps using the R spectroscopy and chemical test to make sure the powder is truly aspirin and not something else. Okay, so in a company, of course, you have to do that. You can't just agree that or believe that, oh, it's coming from the company, you know. That's a problem we had with, as we will come to that, what happened in Kano some years back, where the glycerin was another solvent was used in place of glycerin, and kids were dying. So conduct the assay to quantify the aspirin content and ensure it's between 98 to 102 purity range. Run impurity tests like checking for related substances, maybe salicylic acid as degradation product, while aspirin can break down into that, depending on storage. To ensure impurities are below the allowed limits. You also possibly check the melting point or other specific tests if the monograph includes them. So aspirin monograph historically included a test where you hydrolyze it to salicylic acid and then check the melting point of that salicylic acid. You can also verify loss on drying if moisture content is specified. Only if every single test passes do we accept that batch of aspirin as meeting USP standard. If any test fails, say the impurity level is too high or the identity test doesn't match, then that batch cannot be used for producing USP grade medicine. It would be rejected as non-compliant or adulterated by regulatory standards. Manufacturers incorporate this test into their routine quality assurance. It is not optional, it is fundamental part of pharmaceutical production. And if you don't do this, if FDA or NAFDAC comes in, or if there's a problem, somebody reports, or there's a problem with a batch, and they come in and check to find out that you don't have this, you are in trouble. So each batch of a drug product or ingredient is tested according to the compendium methods or approved alternative methods to ensure compliance before it reaches patients. For pharmacy students and pharmacists, monographs are also relevant. In compounding pharmacy, you might buy your ingredient labeled USP. This indicates the supplier has tested them against a USP monograph and they comply. As a compounding pharmacist, you trust that the standard is the standard, but you might also perform a quick identity check yourself. For instance, verifying the melting point of a chemical or doing a simple chemical reaction test if it is possible for you to do it in the hospital or in your community pharmacy. But these are the kind of things that make you stand out. If it's a teaching hospital, you are supposed to be able to do some of this. So, and if the substance is critical, it is very important that you do that. The USP provides those methods so that even smaller scale practices can ensure quality. So let's say, for instance, a pharmacist making a compounded preparation might confirm they indeed have lidocaine by checking that a portion is made at the right temperature range. A quick identity confirmation will easily suffice, like this, checking for the melting temperature range, or that when dissolved it has the expected pH or specific rotation as per monograph information. So regulatory inspectors during audits of manufacturing facilities or compounding pharmacies will often verify that the tests in the monograph are being carried out How do you do that? You have to have records. So if you don't have your records, there's no way you can prove. And records are very important. They put the data. Those are the things they will look for when they come. And so when we talk about uh, uh, principles of laboratory practice, these are the things we look at. If a company skipped a test or used an inappropriate method without proper justification, that's a serious violation. And the monograph is essentially the benchmark that everyone has to meet. When we teach principles of good laboratory practice, you cannot change anything, any protocol, use anything outside of SOP without justifying why you did it. And this is very, very important. So it's one of the duties, the responsibilities of the study director or the lab manager in this particular case. And remember that the monograph refers you to general chapters for detailed methods. In practice, this means a lab analyst needs to be familiar with those general methods. For instance, if the monograph says conductivity is at maybe 645 or 655 or so, sterility test 721 or chromatographic purity referred to 621, the lab will follow the procedure in chapter 645 or 711 or 621 of USP. This cross-referencing ensures standardization. Every lab is performing the test the same way according to USP's defined procedure. So to sum up, using monographs in practice is about ensuring every batch of drug meets the standard. It's like a checklist a quality scientist goes through. Like it's a checklist the quality scientist goes through, and you probably will be the quality scientist. This guarantees that when you as a pharmacist receive a bottle of a drug, raw ingredient or finished product that says USP on it, you can trust it has been through rigorous checks. It's one of those behind the scenes aspects of pharmacy that protect patient safety and drug efficiency. I hope we are trying to pass the knowledge. Okay. Okay, welcome back. Let's shift gears to a specific aspect of monographs, the solubility description. Often in the description section of the monograph, USP uses certain standard phrases to indicate how soluble a substance is in water or other solvents. These USP solubility terms correspond to exact quantitative ranges. For instance, how many mL of solvent are needed to dissolve 1 gram of substance. It's like a code. Here are the terms and their meanings in plain languages. One is very soluble. When you say that it's very soluble, it means that less than 1 part of a solvent is required to dissolve 1 part of solute. In other words, less than 1 ml of solvent can dissolve 1 gram of a substance. This is extremely soluble. Then you say freely soluble, from 1 to 10 parts of solvent per part solute. So what this means is that 1 to 10 ml of solvent will be required to dissolve 1 gram of a substance. Now another one is soluble. If your substance is described as soluble in a certain solvent, it means that you need 10 to 30 parts of the solvent to dissolve 1 gram. For instance, 10 to 30 ml to dissolve 1 gram. If it is sparingly soluble, it means that you will need 30 to 100 ml to dissolve 1 gram. Slightly soluble, you will need 100 to 1000 parts of the solvent, or 100 to 1 liter of the solvent to dissolve 1 gram. Very slightly soluble is 1000 to 10,000 parts of solvent to 1 part of solute. Practically insoluble is when you need greater than or 10,000 parts or more of the solvent to dissolve 1 gram. So what it means is that you're going to need like 10 liters to dissolve 1 gram. That doesn't make sense, and it doesn't make sense for any practical purposes. Okay, so this ranges assumes a temperature around room temperature. These are conditions. The room temperature in in Africa is around 29 to 30, but in more temperate regions, we are talking about 20 to 25 degrees centigrade, unless otherwise specified. So if you are carrying out this, then you make sure that the temperature you are dealing with is not above 20 to 25 degrees centigrade. So for instance, if a drug is described as sparingly soluble in water, it takes on the order of tens of millimeters of water to dissolve a gram. Not very good solubility. If it is freely soluble, a gram will dissolve in only a few milliliters of water, which is quite good. US monographs often list these terms for water and sometimes for other common solvents like alcohol, chloroform, ether, and so on. Giving a quick reference of the substance's solubility profile. These are common solvents like alcohol, chloroform, and ether. It's important to remember these terms because they give insight into how you might work with the substance pharmaceutically. And that brings us to why solubility matters so much in formulation. So know that solubility is a critical property in pharmaceutics. As a rule of thumb, a drug usually needs to be in solution to be absorbed in the body, especially for oral drugs. So what we're talking about here is we are looking at the bioavailability impact of solubility. So drugs must be, dissolved to be absorbed. Polysoluble drugs USP slightly soluble or worse, or often show erratic or low absorption in the body. So you have to be very careful with this kind of to find a way to sort those kinds of drugs if you are preparing them. How to manage them, if they are really good, if you need them. If drugs don't dissolve well, it may not reach therapeutic levels in the bloodstream, and that's very important. It might have highly variable absorption from one patient to another, or one day to another, and that's not good pharmaceutically, and it doesn't help clinically. So when you see a USP description saying drug is slightly soluble or practically insoluble in water, that's a big red flag for formulation scientists. It means the drug could be challenging to formulate in a way that the body can absorb. In fact, In a context of biopharmaceutics classification system, drugs with low solubility are categorized as class 2 or class 4, depending on their permeability as well. So a pharmaceutical classification or BCS class 2 drug, for example, is one that has low solubility but high permeability, meaning it can cross membranes fine, but it doesn't dissolve easily. So dissolution is the rate limiting step for absorption. Class 4 is low solubility and low permeability, even more problematic. And if you remember your hydrophile lipophile balance, you understand that when you have class 2 drugs, it means that they're hydrophobic. Because if they can easily permeate, if they're permeable through the membrane, it means they are hydrophobic, but they are yeah, they're hydrophobic. Yeah, that's what I want to say. And if they're hydrophobic, it means that they like, they like that they are very soluble in fat, but not soluble in water. But when you have one that is class 4, that is low solubility and low permeability, that gives you more problem. So the FDA uses a benchmark for solubility, and the drug is considered highly soluble if the highest dose strength dissolves in less than 250 ml of aqueous media across the pH range of 1 to 7.5 at body temperature. If it needs more than 250 ml, it is deemed poorly soluble. And if we correlate this with USP terms, many substances labeled sparingly soluble would not meet the dissolve or will not meet the dissolve in 250 ml rule for typical dose. That means those substances might cause dissolution limited absorption in the body. So as a pharmacist, you might notice such drugs often. And when you notice them, you notice that they have special instructions like take with a full glass of water or take with food to help dissolution, or they might come in special dosage forms. For formulators, a poor solubility rating in USP signals that special formulation strategies are needed. They can't just mix the drug into a tablet and expect it to dissolve quickly in the GI tract. Instead, and remember the GI tract is not hydrophobic, it's hydrophilic, it's aqueous. Instead, they might need to micronize the drug. You know what is micronization? You have to find that out because I might ask that in the exam. Formulate it with a solubility enhancer. To give a perspective, it is estimated that a large proportion of new drug candidates, some say 40% or more, are practically insoluble in water. This trend is because many new drugs molecules are large, complex, and not very water friendly. Yeah. This makes the work of formulation scientists very important, and they have to get creative to make these drugs bioavailable. Let's talk about how they do that and some examples of real drugs and their solubility considerations. I think that's a mistake I made earlier. I was telling you that the drug that has high permeability is hydrophobic. No, it's actually hydrophilic. So, oh, sorry. Yeah, I think I was right. Hydrophobic. And yeah, if it is hydrophilic, it will be able to dissolve, but it's not dissolving. But it has, yeah, that's correct. That was not a mistake. Okay, so we're going to look at specific examples of drugs and their solubility considerations. Hang on there. So when you are faced with a poorly soluble drug, Formulators have several strategies to improve solubility or dissolution rate. So one of the things we could do is salt formation. You can convert the drug to a more soluble salt form. For instance, some polysoluble free acids can be formed as the sodium or potassium salt. Typical example is penicillin V. So penicillin V can be formed as the potassium salt, and then that shows huge solubility gain when that is done. Another thing you can do is the particle size reduction. When you increase the surface, when you reduce the particle size, you increase the surface area, and then solubility increases. So, and one of the ways to do that is by grinding the drug into finer particles, which is what you call the micronization or even nanonization. And of course, you form nanoparticles out of them, and this is actually contemporary. You greatly increase the surface area that is in contact with the solvent. And we're talking about the solvent here, we're talking about the gastrointestinal fluid. More surface area can speed up dissolution, and this is like crushing sugar into a powdered sugar, so it dissolves faster in water. So the easiest way to look at this, even though sugar dissolves very fast in water, if you pour sugar granules into water and then pour and drop a sugar cube into water, you notice that the sugar granules will dissolve before the sugar cube will even get dissolved. Both of them are soluble quite alright, but it's a typical example of how micronization functions and why increased surface area increases the speed of dissolution. So you can also use solubilizing excipients. Formulators can add other ingredients that help a drug to dissolve. This might be surfactants, basically soap like, what surfactants? Surfactants are surface active agents. They're able to, a typical example you might know is your detergents. The detergent you use to remove oily stuff. Most of the dirt or the stains you have are actually oily. They are hydrophobic. That's why you can't use water to wash them off. So when you have a surface active agent that has the head is hydrophilic and the long tail is hydrophobic. So they form what you call a micelle, leaving the head to interact with the aqueous surrounding, while the tails that form inside give you what looks like gives a safe haven for the hydrophobic fat, and then they easily lift them up from your dress. That's what they do. So surfactants help you. So they are soap-like molecules that help you weigh the drug and keep it in solution. This is just exactly what it is. You can also use what you call co-solvents, mixing the multiple solvents together to increase solubility. A drug that doesn't dissolve in pure water might dissolve if you have some alcohol or propylene glycol in the solution. So basically what you're doing is that you're increasing the hydrophobicity. When you have alcohol is not as polar as water. Propylene glycol is not as polar as water. So when you add these things to water, you reduce the polarity, and when you reduce the polarity, it helps to accommodate the non-polar components that are refusing to dissolve in water. That's what co-solvents do. So many oral and injectable solutions use a blend of water with other solvent to achieve solubility. Cyclodextrins and complexation are also used. Cyclodextrins are donut-shaped molecules that can encapsulate hydrophobic drug molecules, making them more water soluble. They themselves can also get into water. A drug, we use this kind of thing in separation. As a separation scientist, you can use it in electrokinetography, electrokinetic chromatography. That's where you can use it. You use it when you have a very good cyclodextrin at a critical molecular concentration. It will be able to help you do separation of your compounds. A drug with poor solubility can sometimes be formulated with cyclodextrin to form a complex that dissolves better. For example, the antifungal itraconazole is very poorly soluble, but there's an oral solution where itraconazole is combined with cyclodextrin to help it dissolve. Other things you could do is solid dispersions and novel formulations. A solid dispersion is basically dispersing the drug in a matrix, often a polymer, at a molecular level. So it's not in a crystalline polysoluble form anymore, but rather more amorphous and accessible to dissolve. Techniques like spray drying, hot melt extrusion can create these dispersions. Other novel approaches include lipid-based formulation for lipophilic drug. Put them in oil or lipid that can form an emulsion in the gut or as nanoparticles. You can also do pH adjustment, or form them as prodrugs. You know what prodrugs are? Something that can be cleaved in the body. They are the inactive form, but whatever you attach to it can be cleaved off by enzymes and to release the main one in the body. Do you have an example of a prodrug? Do you know of one? If a drug is ionizable or a base, its solubility can depend on the pH. For instance, a weak base might be insoluble in neutral pH, but quite soluble in acidic environment where it becomes ionized. Formulators can exploit this by adding buffers or formulating the drug in a way that it encounters a favorable pH to dissolve. More drastically, chemists can make a prodrug, like I said earlier on, a temporarily modified version of the drug that is much more soluble, which converts to the active drug in the body. So I asked you to give me an example of one. So a typical example is the antibiotic chloramphenicol, which is a prodrug used in injections because chloramphenicol itself is not water soluble enough. So the key point is that when the USP calls something slightly soluble or practically insoluble, the pharmacy team knows there's a challenge. It's like a heads up. This drug won't dissolve easily, so plan accordingly. As mentioned, a huge number of modern drugs fall in that category. So these techniques are widely used. In drug development, improving solubility is often step one to making an effective medicine. Now, let's look at a few concrete case studies or examples of how solubility issues are addressed in practice. So I've talked about penicillin V before, and so some of the scenarios you might encounter is penicillin V and penicillin V potassium. So the penicillin V is the free acid form. It's very slightly soluble in water, hardly dissolves at all. This would be a big problem if you try to make an oral solution or even expect it to dissolve well in the stomach. So what's the solution? Use penicillin V potassium, the potassium salt of penicillin V. The salt form is very soluble in water. Indeed, clinically, when patients take penicillin V tablets, they are actually getting penicillin V potassium tablets. By converting to the salt, the drug dissolves readily in gastrointestinal fluids, ensuring good absorption. This one is a typical textbook example of salt formation to overcome poor solubility. Another one I want you to read up yourself is phenytoin versus phenytoin sodium. Um, you know, how does this improve solubility? Um, so let's look at some freely soluble drugs. When you talk about phenytoin sodium, you also look at ketoconazole. Ketoconazole is another antifungal that has problem, you know, with solubility. So let's look at freely soluble drugs. If a drug is freely soluble in water, for example, many sources of strong acid or bases like metoprolol tartrate for blood pressure or morphine sulfate for pain, dissolving isn't a problem at all. We put it in a glass of water and it will dissolve quickly. So what might be the concern instead? So the problem you might have in this case is that the test might be an issue or irritation, because now uh, it dissolves so well that you might be able to see that it's bitter or it is sour or it tastes one kind. So highly soluble drugs, if bitter, will make a very bitter solution. For instance, morphine sulfate is very soluble but extremely bitter. So if you are making an oral solution, you have to add flavoring or sweetness to make that taste, or to make it disappear, what we call masking the taste. Or consider a freely soluble drug in the stomach. It might dissolve so far that it could be irritating to the stomach lining. Some antibiotics do this. So for very soluble drugs, formulators might worry less about getting it into solution or more about they worry less about getting it into solution, but they worry more about the consequences of it being so readily in solution. So you see the problem with the word. You don't want the one that is very soluble. You don't want the one that is not soluble. So you have to look for something in between. Whether it's very soluble or not, you always have a problem that you solve, and that's life. From the compounding perspective, a pharmacist loves to see freely soluble because it means you can easily make a solution or syrups without solubility issues. It is easier anyway. From experience, it's actually easier to look for syrup that will mask the test. As a matter of fact, you can actually use the syrup as your base, as part of your base. Practically insoluble drugs require special technique. A drug like glycophyrin, another antifungal, is practically insoluble in water. So the way they used to prepare it in those days was as ultramicronizing it. They used to ultramicronize it, basically making it particles extremely fine, and the order of few microns to increase the surface area. That helps its absorption enough for oral use. And I mentioned ketoconazole, an older antifungal, which is still very effective, works really pretty well. It's practically insoluble at neutral pH. Patients were actually advised to take Conoco with acidic beverage like cola to help dissolve it in the stomach, because acid would protonate it and increase the solubility a bit. Nowadays, new antifungals has been developed with better solubility or salt. Itraconazole, which I mentioned earlier, is so insoluble that it had to be formulated with cyclodextrin for oral solutions and has special solid dispersions in capsules. This example illustrates that when a drug is practically insoluble, pharmacists and scientists have to get clever. Maybe advising patients on how to take it with food or specific drinks using advanced formulation techniques can also help to get it into the body. These case studies highlight a principle. Knowing a drug solubility, often gleaned from the USP monograph description, directly informs how you will formulate, dispense, or administer the drug. As future pharmacists, when you see that description or term in a reference book, it should trigger thoughts about whether the drug needs, say, to be taken with a meal or whether a salt form is available that might be preferable. It's a great example of how something in an official standard connects to everyday practice. Okay, so we are almost getting to the end of this discussion. So this is another part of it. We have talked about solubility, we've talked about identification, we've talked about a whole lot of other things. But now we're going to look at the identification tests. Now there are two types. Is either you are doing the weight identification test, which is the one you do chemical tests in the laboratory, or you do the instrumental method if you have the instruments available. So the very first and what is like the gold standard in the USP is the infrared spectroscopy using the infrared, the IR fingerprint. I have mentioned this earlier on. So we know that the monographs often require multiple ID tests to ensure a substance's identity. So let's talk about the instrumental methods of identification, which are modern and highly specific. So I mentioned the infrared spectroscopy. This is considered a gold standard in many monographs for identifying a pure chemical. Think of an IR spectrum as molecular fingerprint. It shows absorption peaks corresponding to the vibrations of chemical bonds in the molecule and is very specific for a given compound. In USP practice, the usual approach is run an IR spectrum of your sample and compare it to the IR spectrum of an authentic US reference standard of that drug. If the spectra match, especially when you superimpose the standard from the test, you have strong evidence that the sample is exactly what it's supposed to be. It is very unlikely for two different compounds to have the same IR fingerprint, especially in the fingerprint region of the spectrum. In fact, USP has general chapter on spectroscopy identification that basically says an IR comparison with a reference standard can provide conclusive evidence of identity. So you will see a lot of monographs where identification test A is IR absorption, and the test passes if the IR spectrum of the sample is concordant with that of the USP reference standard. Now, what I mean here is that if you superimpose, you won't see any deviations. Would I call it deviation? If you superimpose the two spectra, you will see just one spectra coming out because they are the same. As long as you run them using the same chemical environment, for instance, using the KBr, potassium bromide, and you run it using the same machine, you should be able to get, well, not necessarily the same machine anyway, but if it is run with the same chemical environment, mix it with potassium bromide, and then take the run. Or if you have the raw, the primary standards, or the official standards, you run them together after this and this environment, what you get is going to overlap, and you won't have any difference when you superimpose them. So so it's not a difficult, very easy thing to do. IR requires a little sample preparation, traditionally making a KBR pellet or using a TR detector, but it is quick and powerful. Now, the other thing you can use, but which is less reliable, is the ultraviolet, so UV visible spectroscopy. Some monographs also include a UV absorption test, but it's less specific than IR. Many compounds might have a broad absorbance around a certain wavelength, but it can still be used as a supplementary identity check. A monograph might say, for example, identification B, the solution of the sample exhibits an absorption maxima at 275 nanometer plus or minus 2 nanometer with a specific absorption between 45 and 55. And this is telling you that the compound has a characteristic UV peak at that wavelength and a certain intensity. If your sample meets those criteria, it's another piece of evidence it's the right substance. UV tests are often used when a compound has a distinctive chromophore. A chromophore is, how do I explain it? You should know what a chromophore is. Chromophore is a conjugated ring system. It does not necessarily have to be a ring system. Conjugated system is when you have an alternating double bond and single bond. And alternating double bond and single bond can also be alternating single bond and lone pair of electrons. So when you have something a single bond and you have a nitrogen that has lone pair on it, you're going to have a conjugated system. Or you have a double bond, single bond, double bond, single bond. That's a conjugated system. So the longer the path length, or the stronger the absorption. So it's limited to compounds that have distinctive chromophore. They won't stand. So this kind of compounds will be able to absorb at the UV range. They won't stand alone as proof because different substances can coincidentally have similar UV spectra. That's fine. But in combination with other tests, they help confirm identity. So we can talk about optical rotation as well. Many drugs are chiral. So this is where we talk about optical rotation. When you have optical rotation, so we have chiral compounds. You check their optical rotation, whether they are left-handed or right-handed. And when they are racemic, it means that you have equal quantities of the left and the right molecules. And in that case, even when you do that, you will not get any values. So chiral molecules rotate the plane polarized light, and the direction and magnitude of that rotation is specific to the particular molecule, and it's a non-numeric form. USP monographs for chiral substances often include a specific rotation test. For example, dextrose D-glucose has a specific rotation at about 57 plus 57.7 degrees under specific condition. So the USP monograph for dextrose will say the sample's rotation must be within a narrow range of that value. If someone accidentally had the L-glucose, which is rarely seen, but hypothetically, it would rotate light in the opposite direction. Or if the dextrose were impure or partially degraded, rotation could be off. Another example is menthol. Menthol has two enantiomers. The natural menthol is L-menthol, which has a rotation around minus 50 degrees, whereas the racemic mixture, DL-menthol, would have a net rotation of 0 because the two forms cancel each other's rotation. So a rotation test can quickly tell you if you have the right isomer or if your sample has been adulterated or racemized. It's a simple test, just dissolving the sample in a solvent and using a polarimeter to measure rotation. This is very effective for certain compounds. As a pharmacist, you might record specific rotation being used in identification of amino acid, sugars, alkaloids in quality control. Now, this one is also beautiful, chromatographic identification. You can use TLC or HPLC. I love HPLC personally. Chromatography isn't just for quantifying impurities or assay. It can also serve as an identification method. Many monographs that use HPLC for the assay will also say something like identification retention time. Because the retention time, if you run everything in the same condition, will be the same thing at the same concentration. I know that concentration sometimes can affect the retention time. If it's too concentrated, it might reduce the retention time or something like that. So if you run at the same concentration, the same temperature, using the same solvent, using the same solvent program, solvent and solvent program, probably you're going to get exactly the same thing. Difference might not be much as far as the temperature, especially the temperature, is constant. So the detention time for the major peak in the sample solution corresponds to that in the chromatograph of the USP reference standard as obtained in the assay. Essentially, if you run your sample and the reference standard on the same HPLC system, the target compound should come out at the same time, same retention time, if the sample is indeed that compound. Is a good confirmatory test, less specific than IR, because different compounds could theoretically have similar retention times on a given column, but in combination with other data, it's useful. Thin layer chromatography is an older method that is still used in some cases for ID, especially in poor resource countries like Nigeria. You can actually use thin layer. You spot the sample and the reference on a plate, develop it, and see if they travel the same distance. The retention factor or rotation factor value will be the same thing and show the same color reaction with detecting spray. TLC might be found as an identification test in some older monographs of herbal products. So why IR, UV, optical, rotation, chromatography are the big ones. Occasionally, other instrument tests show up. For instance, Raman spectroscopy, near spectroscopy can be used for identification, especially for rapid ID of materials without much sample prep, like in warehouse scanning through packaging. Mass spectrometry and nuclear magnetic resonance are typically not routine monograph tests. They are more specialized and expensive, but some modern monographs for very complex molecules like large peptides or biologics might reference them, or they might be used in certain cases by manufacturers for identification. However, for classic small molecule drugs, you rarely see MS or NMR as the official ID test because USP tries to stick to widely accessible methods, which is very good. Instrumental methods are favored in industry because they are quick, specific, and reliable, but they require instruments and often the use of reference standards for direct comparison, particularly for IR and chromatography. This is why USP also supplies these reference standards to labs. When reading a monograph, you will see something like USP drug name, let's say USP XRS under reagents, which implies you need that standard to do the ID or assay properly. The take-home message here is instrumental ID tests give a high degree of certainty. If the IR fingerprint matches and the optical rotation is correct, it's extraordinarily likely you have the right substance. That said, pharmacopoeias often still include one or two simpler weight chemistry tests as well, which we will talk about next. partly for historical reasons and partly because not every pharmacy or lab around the world may have the R spectrometer on hand. So we're going to talk about that next, especially for poor resource countries like Nigeria or Africa generally. So we're not going to waste so much time on this. We're going to fly through them. Um, Before the modern instruments were common, pharmacists and chemists relied on weight chemical tests to identify substances. So these are basically specific reactions that produce a visible result: color change, precipitate, gas, and even a particular functional group or ion, to find out if a particular functional group or ion is present. Many of these classic tests are still documented in pharmacopoeias and sometimes appear in monographs, especially when the test is simple and effective. Let's just look at a few examples and then you can look for some other for yourself. Precipitation reactions for ions. A typical example of that is the test for chlorides. If a substance is supposed to be a chloride salt, for instance, we're talking about ephedrine chloride or potassium chloride, a clinical or a classical test is to add a few drops of silver nitrate solution. If the chloride is present, you get a white cloudy precipitate of silver chloride. To confirm it is chloride or not some other halide, you then check that the precipitate is insoluble in nitric acid but soluble in ammonia. Those are distinguishable features of silver chloride, for instance. Silver iodide gives a yellow precipitate that doesn't dissolve in ammonia. So USP's general chapter on identification test describes this chloride test. In a monograph, it might appear as add silver nitrate to a solution of the sample. Curdled white precipitate is formed, which is insoluble in nitric acid. If you sample passes that, it indicates chloride. Similarly, there are precipitation tests for other ions. For example, sulfate is tested by adding barium chloride to produce a white barium sulfate precipitate, which is insoluble in acid. Calcium can be tested with oxalate to give a white calcium oxalate precipitate. These tests confirm the presence of specific ions, anions, or cations that should be in the compound. So I wanted to look at other things like color reactions with ferric chloride for phenols and gas evolution or flame tests for yourself. These things are no longer properly used. There are a combination of reactions, plus physical constant. So this is a clever approach that could be used sometimes where a chemical reaction isolates the products, which can then be identified by physical property like melting point. We saw a hint of that in the aspirin case. After precipitating salicylic acid from aspirin, you can filter it and measure its melting point, and that should be between 158 and 161. If it's very accurate, it should be 159. If the melting point matches the known MP of pure salicylic acid, that strongly confirms that what you precipitated was salicylic acid, meaning your original sample was indeed aspirin, because few substances would produce salicylic acid under those conditions. This kind of two-step test create a derivative that check melting point. Was a common one in older pharmacopoeias. Okay, it's very specific and cheap to do, but it takes a bit more time than modern methods. Still, it's a great educational example of combining a chemical reaction with a physical constant to nail the identity. Mm. So we just have to say that chemical identification tests are part of the toolkit. They connect back to basic chemistry, something as simple as mixing solutions and observing reactions, which is pretty neat. As pharmacists, understanding these tests gives insight into why certain reagents um, cause changes in drug solutions. For example, why iron supplements shouldn't be taken with tea. Tannins and ions form dark complexes, a bit like a chemical test happening in the gut. It all interlinks. If you have questions, you can drop them in the comment section actually, and we can attend to them. So now all identification tests must pass, and I think I've talked about this like 2 or 3 times. So we have to bear that in mind. But it's worth stating clearly in an official standard like USP monograph, all the listed identification tests must be passed for the material to be confirmed as the correct substance. Identification tests A, B, C, D, if they are all listed, then you have to do all of them. And they have to pass all of them. The compound has to pass all of them, otherwise, it's not the compound. Identity is essentially a binary criterion. It's either confirmed, it is not, or it is not. And there's no almost identified. You understand what I mean? It's either you confirm it or it's not. So what does this mean practically? Let's say a monograph has 3 ID tests. I has spectrum matched, B a chemical color reaction, and C perhaps chromatography retention time match. The sample has to pass A, B, C. Even if one of those gives a result that doesn't match the requirement, the sample is considered identification failed. Let me say that again. If just one of those gives a result that doesn't match the requirement, the sample is considered that the identification has failed. In regulatory terms, if a material fails an identification test, is treated as misidentified or adulterated, you cannot use that material in manufacturing. It shouldn't be dispensed. There is no acceptable level of non-identity. It's a yes or no gate. It's either yes or it is not. So let's say you have IR spectrum matches USP aspirin and ferric chloride color test for salicylic, and then you have perhaps a melting point of the salicylic acid. Only if all are positive do we confirm yes, this is aspirin. So if this doesn't work, then it doesn't work. So you have to consider it like checking someone's identity with multiple factors: photo ID, fingerprint, and security question. These are the things you do if you have passed through the immigration in some countries like the US, or when you're even going to get visas. You have to drop your your photo ID, you do fingerprint. Next time you come, they have to match all those. If you don't, you are sent back. Anybody that comes with something that is different will be sent back, that is not you, even if the name is the same. So if one doesn't, something's wrong or something is wrong. Similarly, a counterfeit or wrong chemical would have to fool all the tests to slip through. Highly unlikely. Maybe it could coincidentally have a similar IR fingerprint as the intended drug, which is rare, but let's imagine. But then, if it doesn't produce the right color reaction, you see what it is. You say, okay, that's it. And that will help you eliminate this rogue compound. We can also have two compounds, two substances that might coincidentally both form a white precipitate with silver nitrate. Any chloride salt will, but they won't both have the same specific rotation aspect spectrum as the true drug. So the combination provides high confidence, and that's why the monograph goes for that. From a quality control perspective, when an identity test fails, there's no debate or a test to see if it is within some limit. It's simply not the correct material. That's it. And this all or nothing approach is unique compared to other tests like assay or impurities, which has acceptable ranges. Identity is absolute. Let's bring back the aspirin tablet example, illustrate multiple ID tests working together. Imagine you have a tablet that's supposed to contain aspirin. I've already talked about told you about this, the IR specific test or an extract of the tablet or find that IR matches the USP aspirin reference standard, which is good. That indicates aspirin structure is present. You also perform the ferric chloride color test after hydrolyzing a bit of the tablet in acid, which would liberate the salicylic acid if aspirin is there. And you add ferric chloride and get a purple color. Okay, that's great. That indicates the salicylic acid is present consistent with aspirin. Perhaps you also run a quick thin layer chromatography and see that the main spot from the tablet has the same RF at a reference aspirin solution. And maybe after spraying with a reagent, it gives same color, another confirmation. If all those tests align, you can be extremely confident that the tablets contain aspirin. If one of them failed, say the IR didn't match, you will be immediately concerned that something is wrong. Maybe the tablet has acetaminophen by mistake instead of aspirin, which actually happened historically in some mix-up cases. Even if the chemical test gave um, purple, which acetaminophen wouldn't, or hypothetically, a non-matching IR would stop it, or vice versa. So for students like you and future pharmacists, we hope that you will be. The lesson is never ignore an identification failure. In the lab, if identity fails, you don't go, well, the assay was okay, so maybe it is fine. No, identity trumps all. You either positively identify the material as correct or you treat it as the wrong material. This is the fundamental to patient safety. Using the wrong ingredient or mistaken identity of a drug could be dangerous. So the standards are appropriately strict here. Let me repeat. The standards are appropriately strict here. So to wrap up identification tests, they are performed to ensure authenticity of the material. Multiple tests provide a safety net and absolutely all must pass. ESP and other pharmacopoeia built in this redundancy to protect against any single point of failure. It is like having multiple locks, all must be unlocked, test passed for the standards to be cleared for use. So we'll talk about some of the constants that could be used or can be listed in the monograph. We'll talk about the melting point, is a quick check for purity, refractive index, specific gravity for liquids, optical rotation. We have mentioned this a couple of times. So they're beyond the high-tech instruments and chemical reagents. Quality control also leans on good old physical constants or substances. These are properties like melting point, as I mentioned. They might sound like things from a chemistry textbook, but they are very useful in pharmacopoeia standards and real-world quality control. Let's go through them one by one. So melting point or melting point range. Sometimes it's not, you can't get it sharp. So many pure solids have a characteristic melting point. It's quick to measure. You just heat a small sample and see the temperature at which it turns from solid to liquid. In pharmacopoeias, you will often see something like melting range. In a monograph description or test, you also see that, okay, because the pharmacopoeia uh, uh, of course, the pharmacopoeia is made up of the monographs mostly. So a pure compound usually melts sharply at its specific temperature over a very small range. If there are impurities, the melting point typically drops and broadens. So if you have a drug that is supposed to melt at 150 and your sample melts at 140, 148 range, over a wide range like that, that's a hint something of maybe it's impure or it's even a different substance. Melting point was historically a primary identification and purity check. Well, nowadays we look at it as a supplementary test, but it's still included for many monographs, especially older ones and the experienced. For example, cocoa butter in the NF has a defined melting range. Another example, if you isolated salicylic acid as in the aspirin test, checking that it melts at around 159 confirms its identity. So melting point is simple but powerful. It confirms identity to an extent and indicates purity. Boiling point also is another thing, um, which I won't want to go to, but it's also analogous to melting points, can help identify a liquid and check its purity. In USP, you don't see boiling point used as often as melting point, except for things like solvents or essential oils. For instance, USP ethanol might be checked via specific gravity mostly, but historically, one could check its boiling range to ensure it's correct. On the purity of something like benzene or acetone, it could be assessed by boiling point. If a liquid boils way off the expected temperature, likely not what you think it is, or is heavily contaminated. So let's talk about optical rotation. I think I mentioned this before, so let's not go around that again. And the next one is refractive index. So refractive index measures how light bends when it enters the substance, usually a liquid. It's very sensitive to liquid composition. Many monographs for oils, solvents, or liquid chemicals specify an RI at a certain temperature, commonly 20 degrees centigrade or 25. For example, glycerin has an RI of 1.473 at 25 degrees centigrade. If you had a bottle of glycerin and wanted to know if it is up to standard, measuring the RI is one quick check. If it got, say, 1.46, that suggests the glycerin might be diluted with water or another solvent, because pure glycerin should be higher. So refractive index can catch even subtle adulteration, since adding a bit of water to a glycerin shifts the RI measurably. Essential oils have characteristic RI ranges also. For instance, peppermint oil or clove oil have often list RI and optical rotation as identity checks. Adulterations with cheaper oils changes these values. In a pharmacy context, one might check the RI of a compounded solution like a concentrated sugar syrup to ensure concentration is correct. But in manufacturing, digital refractometer are widely used to quickly assess purity and concentration of liquids. Finally, let me talk about specific gravity, which is also called relative density. This is basically the density of a substance compared to that of water. ESP uses specific gravity for many liquid ingredients. It is simple. You measure how heavy a volume of the liquid is relative to the same volume of water. For example, concentrated sulfuric acid has a very high specific gravity. Glycerin is about 1.25 ethanol, 95%. 95% ethanol is about 0.816 at 20 degrees. So a monograph may say something like specific gravity 1.18 to 1.19 at 25 degrees centigrade for a solution. If your sample's density falls outside that range, something is wrong. Perhaps it's diluted or impure. Specific gravity is extremely useful as a quick check because you don't need fancy equipment. Just a pyranometer or hydrometer or a digital density meter. Pharmacies traditionally use specific gravity to check alcohol strength or to measure out quantities of liquids by weight. In quality control, it can confirm concentration. For instance, a syrup USP has a specific gravity requirement to ensure the sugar content is correct and not too watery, which could allow microbial growth. Specific gravity is also used to confirm that something like a supposedly pure oil hasn't been mixed with another an adulterant, something different, something like different oil that has different density. So a mismatch could signal adulteration. So in summary, what we're trying to say is that physical constants are fundamental properties that serve as quick identity or purity checks. A correct melting point builds confidence that a solid drug is what it should be and fairly pure. Optical rotation RI ensure the identity and purity of liquids or chiral compounds, catching things like wrong isomers or dilation. Specific gravity confirms concentration of solutions and purity of liquids. This test has been part of pharmacopoeia standards for ages and remain in use because they work well and require minimal resources. It's a nice reminder that even as technology advances, the basic principles of chemistry and physics remain very relevant in quality control. So, getting to the end of this, like I said, I think one or two more slides and we'll be done. So, I want to bring it all together. Let's talk about some quality assurance best practices in the context of this official standard. Whether you end up working in a pharmaceutical company, compounding pharmacy, or even indirectly in roles like regulatory affairs, understanding and applying these standards is crucial. Here are some key practices and insights. Always adhere to compendium methods and specifications. If a drug's monograph specifies a test, that test should be performed. or a validated equivalent if allowed, and the result must meet the acceptance criteria. Skipping tests or using unproven methods can lead to serious quality issues. If a drug's monograph specifies a test, that test should be performed, or a validated equivalent if allowed, and the result must meet the acceptance criteria. Skipping tests or using unproven methods can lead to serious quality issues. Companies often incorporate all USP monograph tests into their internal QC control protocols. And this might sound obvious, but it's a cornerstone of quality assurance. Follow the official recipe exactly to verify quality. Use reference standards and proper controls. To confidently say a test passes, especially instrumental tests, you need to compare to a known standard. That is why USP reference standards are used for things like IRHPLC and so on. Also, run appropriate blanks and system stability tests as described in general chapters. This is very important with HPLC. And in fact, not just HPLC, every instrumental method. You have to rule out noise, and that helps you to get things, to make sure that what you're doing is right. You might see a noise, might see, for HPLC for instance, you might see noise coming from a bubble, and you think it is a substance or a solute. Best practice is that every analysis should be backed by evidence that the method worked correctly. For instance, HPLC, the reference standard should give a proper peak at expected retention time, and this ensures your test results are reliable. I want to talk about proper peak. It should be symmetrical, not asymmetrical. A peak that is telling whether it's dragging or telling, it won't be what you're looking for. It should be a symmetrical peak and shouldn't be too broad, except this is what it has been known to do, or the standards are shown have been shown to have broad peaks in the given condition. Leverage simple tests for quick checks. A good QA practice is to use the simple physical chemical tests as initial screens. For example, if a new batch of chemicals come in, the technician might quickly check its melting point, and perhaps a quick ID test like IR or a spot test, even before doing more time-consuming access. If something is off, you catch it early. Think of the earlier peanut oil example. If the USP monograph says peanut oil's specific gravity at 25 degrees should be 0.912 to 0.920, and the batch you got is 0.900, you immediately suspect it might be adulterated, maybe someone diluted with lighter oil. You wouldn't want to go through full microbiological assay tests only to later find it was the wrong oil. So quick checks like specific gravity, RI can save time, and then act as red flags. Understand the context of tests to troubleshoot issues. A QA professional should know why each test is there, not just treat it as a checkbox. For instance, if the assay of the drug is low, one might also check the impurity profile. Maybe the drug partially degraded, so impurities should be up, the API will be down. If an ID test fails, one should halt and investigate if perhaps the sample was mislabeled or the test was done incorrectly, but never ignore it. If a dissolution test fails for a tablet, it might indicate a formulation problem affecting bioavailability. By understanding the purpose of each compendial test, you can better respond when something doesn't meet the standard. So integrate the compendial standard into SOPs. companies will usually build their own testing protocols, referencing it using a USP method. As a best practice, these SOPs should be kept updated when USP updates a monograph or a general chapter. Pharmacopoeias are not static. They get revised. In fact, they get revised very often, sometimes more than once a year. Or often you have updates that come up within the year, and then they incorporate that when the next version of it comes up. Quality units must stay abreast of changes. For example, if USP tightens an impurity limit or introduces a new method for an existing test, the company should adapt. Similarly, pharmacies that do compounding should ensure they use current guidelines. Training and familiarity is very important for pharmacists and technicians. Being familiar with the reading and interpreting monograph is essential. It might seem daunting with all the jargons, but with practice you can quickly pinpoint what each test is about. Best practice is to train staff not just on how to do something, but why they do it. For example, explain to a compounding tech that we measure the specific gravity of our prepared cough syrup to make sure that the sugar concentration is right by USP. And this ensures the syrup will be properly preserved and have the right viscosity. This ties the everyday task back to an official standard and the quality reason. Official standards are a foundation, but the real world throws curveballs. Maybe an excipient in a tablet causes a USP assay to have an inference. Companies might have to validate a modified method, or a new impurity is discovered that the monograph doesn't explicitly list. The quality team should address it, perhaps report it, and get the standard updated. Best practices involve not only following compendial standards, but also going above and beyond when needed to ensure quality. Always remember that monograph is a quality contract. A phrase often used is that pharmacopoeia monograph is like a quality contract. It's an agreement that if the supplier provides material meeting all these criteria, the material is acceptable. Conversely, if you are the purchaser, you are the pharmacist or manufacturer using the material, you have the right to reject it if it doesn't meet this criteria. Everyone knows what the expectations are. Excuse me. Embracing that mindset is the best practice. It keeps suppliers accountable and gives purchasers confidence. Suppose you are in charge of the quality of pharmaceutical ingredient distributor. You get a new batch of acetaminophen powder. First, what do? Verify the certificate of analysis from the supplier. So you want to be sure that the certificate analysis from the supplier has all the USP tests listed and within spec. So then you go ahead and do your own identity test, maybe an IR or a melting point, because acetaminophen has a known MP of around 169. Check a quick assay or potency, maybe a rapid DHP run, or even titration if available to confirm it's 100%. Ensure impurities like para-aminophenol, a non-impurity in acetaminophen, are low by the method USP suggests. If all that looks good, you release the batch for sale or use. If anything is off, you quarantine it and investigate. By consistently applying the official standards in this way, you maintain a high level of quality assurance It protects patients, it protects your company from regulatory trouble, and it upholds the trust in the USP label. For you as future pharmacists, even if you are not in a lab, knowing that these best practices are followed can give you confidence in, in the medicines you dispense. And if you compound or do any small scale manufacturing, you'll be the one to implement QA steps like checking identity and potency of ingredients as guided by this standard. Finally, I want to tell you that standards protect public health. It's very, very important for you to know that. So we've covered a lot of ground from what a monograph contains through solubility, identification tests, physical constants, and quality practices. Let's take a step back now and appreciate why all this matters on the big picture level. The ultimate goal of official Marcopio standard is to protect public health by enforcing that every batch of a drug meets certain criteria. We vastly reduce the chances that patients receive medicines that are subpotent, contaminated, or just the wrong thing altogether. It creates a safety net for the drug supply. Think about it. When a patient takes a tablet, they probably never consider that someone tested that tablet to make sure it dissolves properly in 30 minutes, or that the active ingredient was identified by IR spectrometry, or that the raw powder's melting point was checked. But because all those tests happen behind the scenes, patients can simply trust that the tablet will work as expected. This trust is the cornerstone of medicine, and pharmacopoeia standards are a big reason that trust is justified. For you in year 3, PharmD students, very soon you will be called doctor in the next 3 years. Understanding these standards is like peering under the hood of the medication quality system. It connects the dots between your basic science courses, chemistry, pharmacology, and practical outcomes. For example, you learned in chemistry why a precipitate forms. Here is seeing that the principle applies to identify a drug. You learned about chemistry, chemical structures. Here you see an IR spectral fingerprint being used to confirm a structure. You study dissolution and bioavailability in pharmaceutics. Here you see dissolution tests in monographs, ensuring a tablet will release the drug properly. You have heard of potency and stability. Here you see assay ranges and purity limits, quantitatively defining those. By mastering pharmacopoeia standards, you gain insight into how each pill or injection in a hospital or pharmacy is backed by rigorous testing. This knowledge will make you a better pharmacist because you will understand why storage conditions matter, since a monograph might say protect from moisture for a reason, or why you cannot substitute one sort of a drug for another if the monograph and standards differ, or simply how to interpret a drug label that says USP or BP on it. Moreover, these official standards have a global aspect. The USP's mission includes improving global health through public standards. When a standard is created or updated, USP invites input from scientists around the world. It is a continuous process of scientific refinement aimed at responding to new information, like discovering a new impurity or better test methods. As medicine advances, think of a new biotech drug, advanced therapies. The pharmacopoeias evolve as well to create appropriate standards for them, always with patient safety as a priority. In essence, pharmacopoeia standards ensure consistency. So one company's drug and another company's version meet the same minimum quality. Safety impurities are kept to safe level, no toxic substances, and we're talking about efficacy. Of course, when you talk about biosimilar, you have generics. The drug contains what it should and will deliver it properly. So this system prevents a scenario where you say one manufacturer's 100 milligram tablet actually has 50 milligram of drug, which could be disastrous because that tablet wouldn't pass the USPSA requirements. Or it prevents someone from selling amoxicillin that's actually something else because it wouldn't pass the identity test. To wrap up, these standards may seem technical and detailed, and they are, but they are fundamentally about protecting patients. As pharmacists, we are advocates of our patients' well-being. The well-being of our patients, and part of that is ensuring that the products we dispense are of top quality. Official pharmacopoeia standards are one of our strongest tools to guarantee that quality across board. By enforcing and adhering to these standards, we uphold a crucial aspect of public health, that every medicine is as safe and effective as should be. So at this point, I would ask you if you've got any questions. So that concludes our exploration of official standards for pharmaceutical chemicals and formulated products. We have seen how monographs act as blueprints for quality, how solubility and other properties play in the formulation and testing, how identity and purity tests ensure we have the right stuff. I hope this session has demystified the role of pharmacopoeia standard and shown you how it connects to what you do as pharmacists. Pharmacy students is a lot of detail, but remember the big picture. These tests and specifications exist so that when you give a medication to a patient, you can be confident in its quality. Thank you very much for getting to this point. I've enjoyed working through this material with you in an academic yet hopefully approachable way. If you have any questions, I'll be happy to answer them now. Feel free to ask about anything in the comment section, whether it's clarifying a concept like a specific test or how these standards are used in pharmacy setting, or even historical tidbits about how these standards came to be. Don't hesitate. We're all here to learn and ensure we are prepared to uphold these quality standards in our future careers. Thank you very much.